week 4 live interaction session of the Environmental Biotechnology course. So yes, so these are the concepts we'll be covering today. So first we will be studying how the integration of different omic approaches helps us in the environmental biotechnology. Then we'll be studying about the classification of microbes and their quantification. Then finally we'll be learning about the prokaryotic cell cycle and how we can model the microbial growth based on the concepts related to microbial growth. So coming to the
I can't see the screen.
sorry,
I can't see the screen.
okay, wait. Is it visible now?
Yes, it is.
Yeah. So omics related approaches can be classified into uh, different omic based approaches, which can be classified into metagenomics, transcriptomics, proteomics, and several other omic based approaches are there. So using these omic based approaches, we can study the structural and functional module of different organisms. So why it is so necessary to study the genome, or any other entities related to omics? Because studying genomes will give us the expression profile of a particular microorganism. So this expression profile will change with respect to the environment present, the microorganism present in a particular environment. So simply by studying the expression profile and kind of DNA material present in a microorganism, we can tell the kind of changes happening in a particular environment, because the microbes will adapt to the environment. So based on that, the expression pattern will change. So by utilizing omic based approaches, we can study the expression pattern and tell us how the microbe behave to a certain environment. So here specific challenges. Usually these omic based approaches are used for single organisms. So if we want to use this approach for multiple organisms present in a natural environment, a lot of challenges are there. So that we'll be studying in the upcoming slides. So what are those to address those challenges? The research should be more of theory or process driven. It should not only depending on tools. Yes. Yes, to address the problems of environmental biotechnology, the approach should be more of a process driven. Like there are a lot of tools are available, like omic based tools and mathematical tools are available to solve the problems in environmental biotechnology. So only with the tools we cannot come to a conclusion. So the tools will give us some results. Only if you are relying on some process driven research, then we can come to a interpretation. So the emphasis should be more based on process. We should understand the process. So based on our understanding, we should apply the tools available with us, like omic based tools, or any other mathematical tools. So the first and foremost thing is we have to rely on process driven research. Yes, the first and foremost thing here is like we have to rely more on the process driven research. And one more challenge is here, like microbial ecology is completely different from macro ecology. Do you,
Sir, screen is not.
screen is not visible. Is it visible now? Yeah, is it visible now?
Yes, sir.
Yeah.
Yes, sir. Yes, sir.
Yeah. So we cannot compare the microbial ecology with macro ecology, because the microbial communities in space will vary with respect to different parameters, and also it is related to relationship between in different species and the same species itself, and also their environment. So we cannot compare microbial ecology with macro ecology. So this is a challenge in studying the microbial communities. So in order to study a particular microbial community, we have to consider the spatial and temporal scales, which means the amount of space occupied by the microbial community, and also the environment, the kind of environment it is in. So usually the microorganisms has a size of have size of in the range of micrometers, but microbial aggregates can exceed above 1 millimeter. So we have to be aware of these scales in order to understand them. So if we want to model the organisms, we should know how the organisms are spatially distributed and what are the factors affecting the distribution of microbes. So we can study these things through modeling, because it involves different transport processes, and it also considers heterogeneity. Here transport processes refers to the transport of nutrients and outcome of products. Then here and heterogeneity refers to the population, what all the different species present in the microorganisms, microbial communities. It refers to heterogeneity. If different species are present in a particular space, then we can call that population as heterogeneous. So how we can study this microbial community through integration of different fields. So we can rely on different materials, modern materials, to create a microbial niche. So this niche will support the growth of different organisms. So those materials can be uh, classified as membranes, ceramics, semiconductors, conductors, and nanomaterials. These membranes, ceramics, are those things are used in wastewater treatment, and the semiconductors, conductors, nanomaterials are also used in wastewater treatment, but the processes are different. These membranes are used in membrane bioreactors, and these ceramics are used as microbial growth supports, whereas these semiconductors, conductors, and nanomaterials are used in the fuel cells, microbial fuel cells. These microbial fuel cells can be used as for the treatment of wastewater, and also for generating energy. Yes, as mentioned here, mostly these materials are used for supporting the growth of biofilm. So coming to the modeling part, we can model the growth of a particular organism based on the flow of electrons. So in order to study the flow of electron, we should understand the different entities present in the microbial growth, which includes mass transport, the transport of nutrients and the product out of the cell, and kinetics, what is the time taken to undergo these reactions, and also we need to rely on other engineering principles, like growing those microorganisms in reactor. So we need to have a basic understanding of these all concepts. So why we should rely on modeling and simulation? So simply, if we model a system, yes, what is the need for modeling? So only if understand the kinetics and mass transport of a microbial growth, we can model a system. So if we model a system, we can create engineered system for different applications. It may be for wastewater treatment, or for synthesizing industrially valuable chemicals. So we can create an engineered system by modeling the microbial system, and we can develop a cost-effective method to employ. So in addition to that, we can also manage the microbial communities and use it for our basic or specific needs. So by modeling the microbial growth, we can support them, like we can study the metabolism of microorganisms, and we can support their growth. So simply, if we take care of the needs of microorganisms by modeling them, it will work for us. So these are the challenges which are involved in the studying the microbial communities. So the first and foremost thing is the complexity of microbial system. So microbes can grow faster, and also different microbes can coexist together based on the synergism between the organisms, or they can be separated apart in space based on the antagonism exhibited between the microbial communities. So this complexity creates a challenge for studying the microbial communities. So the next thing is only few strains can be cultivated. Like there are almost 5 lakh strains are there. Out of that, only few microbial strains can be cultivated in the laboratory settings. That is a major problem. So one more thing is microorganisms can evolve quickly. So if you want to study a particular microorganism and we are extracting the like genome of a organism and studying that genome will change due to the evolution of the microorganisms. So even if you study a genome of a particular organism, it will be of no use after some point of time. So that is a problem in studying the microbial community. That is one of the problems in studying the microbial communities. And in the case of different environmental matrices, even if we isolate some organism, due to the nature of the environmental matrices that the organism is present in, the extraction of genome and studying the microorganism will be so difficult due to the interference of other different chemical components and physical components present in the environmental matrices. So this creates a major challenge. So in order to understand the microbial communities, we have to go through all these challenges first. So how can we quantify microorganisms? So why we need to study microbial groups and quantify them? So in order to develop technologies for the treatment of water, wastewater or solid waste, so that is the major application as we already discussed. So only if we study especially in the case of pathogenic organism, if we study the microbial behavior, we can prevent the spread of the microorganism through water, soil or vector. Based on the nature of the microorganism, we can prevent the spread of those microorganisms. Ultimately, we can prevent the disease spread. So that is on need for identifying microbial groups and to quantify them. And again, one more application is monitoring and controlling different environmental systems. So for all these applications, we need to identify different organisms and quantify them. So in environmental microbiology, we need to take care of certain aspects. The first and foremost thing is to classify and identify microorganisms. How classification and identification differ? So classification refers to classifying a microorganism what can we say? Classification refers to bringing a microorganism to a certain group of microorganisms. So this refers to classification. Identification refers to the particular microorganism identifying the organism and determining to which class of organism that microorganism is belong to. So that refers to identification. And the second thing is we need to study how microorganisms are interacting with the physical, chemical and biological entities present around them, which are nothing but biotic and abiotic factors, and how they are interacting with the macroorganisms present in a particular environment. So that also covered by environmental microbiology. So majorly microorganisms can be classified into bacteria, archaea. So these group of organisms come under the class of prokaryotes. Prokaryotes means the nucleus is not well defined. Nucleus and the cells are not membrane bound. So those organisms are classified as prokaryotes. And bacteria and archaea come under the class of prokaryotes. Then other than that, these are like eukaryotic organisms, which includes microscopic fungi, microscopic algae, protozoa, and viruses, metazoa, and cysts of helminths. So we'll be studying in detail about all these organisms in the upcoming slides. So coming to the
last slide.
Sorry.
In last slide there was chemical interaction it was talking about. So which type of chemical interaction it was here?
Wait. Last slide, right?
Last slide.
Yes, let me. Here you are asking, right? Sorry. Here you are asking, right?
Yes, sir. Last second line. Yes, sir.
Yeah, chemical interactions. Yes, actually, the pH of a particular environment depends on the chemical nature, isn't it? Based on H plus and OH ions. So that refers to chemical interactions. Based on the environmental pH, you know that microorganisms can survive in a particular pH range, like 2 to 9. So that depends on the chemical environment, what kind of chemicals present in a particular environment. So that comes under chemical interaction. Other than that, if some toxins are present, so sometimes those toxins can induce the microorganism metabolism of microorganism. So in that case, it also comes under the chemical interaction.
So like nitrogen cycle, carbon cycle, they also might be included in this?
Yes, of course. Yeah, wait. Someone wants to join, let us.
Full boy party.
Yes. Yes, you are asking some question. Yeah. Biogeochemical cycles. We were talking about biogeochemical cycles, right? Like nitrogen cycling, isn't it?
Yes, carbon cycle and
Yes, those interactions are also coming under chemical interactions.
all those are chemical
Yes,
interactions with
all those are chemical
environmental.
interactions. Yes. Your doubt is clear? Your doubt is clear? Hello.
So and physical interaction.
Yes, physical interaction refers to temperature.
So
Temperature.
Hello.
Yes, yeah. Hello. Yeah, can you hear me? Yes, sorry for the interruption. You were asking about physical interaction, right?
Yes, sir.
Yes, yes. It refers to the surrounding temperature and also the what can we say, the base to which microorganisms are attaching. So those interactions come under the class of physical interactions. So
Sir, some example.
for say like microorganisms will for attaching in a smooth surface. Usually microorganisms cannot attach through on a smooth surface. So in order to like facilitate their attachment, they will secrete some surfactants. Have you heard about it? For facilitating biofilm formation, the microbes will produce some chemicals that come under the class of physical interaction, because usually microorganisms cannot what can you say, grow on a smooth surface. To make a surface rough, it will produce some chemicals. So it is an example of physical interaction.
So, sir, quantum sensing is physical or chemical?
It comes under chemical interaction.
Okay, so when they are forming biofilm, they are also using chemicals, right? So how can it be in physical interaction?
For attachment, they are actually yes, they are producing certain chemicals, but those chemicals are not what can we say, facilitating attachment. Ultimately they are providing physical interaction, isn't it? Ultimately they are providing physical interaction only, right?
With the surface.
Yes, yes. Ultimately it is facilitating attachment.
Okay.
Ah,
Thank you, sir.
yeah. So these are the key aspects of environmental microbiology. We already discussed about these things. Yes, we were discussing about the prokaryotic organisms. Actually, prokaryotic organisms rely on different energy sources for their growth. So, if prokaryotic organisms rely on chemical compounds, we can call the mode of nutrition as chemotrophic. Or if they rely on light, we can call the mode of nutrition as phototrophic. And based on the kind of substances a prokaryote is using, we can classify the mode of nutrition as heterotrophy and lithotrophy. If an organism uses organic substances for their growth, then we can call those organisms as heterotrophic organism. In the case of inorganic substances, if an organism is using inorganic substances, we can call those organisms as lithotrophic. So based on the source, we can classify the organisms into chemotrophic, phototrophic, or heterotrophic, or lithotrophic. So in prokaryotic growth, other different physiological characteristics also involved. The carbon source, even though already we discussed, the carbon source can be carbon dioxide or any other organic compounds. If the carbon source is carbon dioxide, we can classify those organisms into autotrophic nutrition. In the case of organic compounds, we can classify them as organotrophic. organisms, and the temperature also varies for prokaryotes. The prokaryotes, they can grow in a wide range of temperature, that is from 0 degrees Celsius to 100 degrees Celsius. Usually normal microbes cannot grow in the extreme temperatures, that is in very low temperature or very high temperature. But in the case of what can we say, RK, they can grow in these extreme conditions. They are called as like extremophiles, as we all know. And the pH also varies. Like the microbes can, especially prokaryotes, can grow in the range of 2 to 9. And also here also the microorganisms, specifically RK, can grow at the extreme pH levels. Not all prokaryotes can grow, but RK can grow at the extreme pH level. In the medium pH, other bacteria can grow. Different classes of bacteria can grow. So if we take prokaryotes, so the oxygen plays a major role. How can anyone explain, like, what is the role of oxygen?
So facultative anaerobes or
Yes. What is the role of oxygen?
so it helps to grow and reproduce.
Sorry.
Or not. It helps microorganisms to grow and reproduce.
Yeah. Specifically, in which way? If we talk about metabolism, what is the role of oxygen? It acts as a electron acceptor, isn't it? In the microbial metabolism, it acts as an electron acceptor. Basically, what we have studied, microbial metabolism or redox reactions. So in that redox reactions, electron transfer reactions are prominent. So for electron transfer reactions to happen, we need a certain electron acceptor. Here oxygen plays the role of electron acceptor. So based on the interaction of microorganisms with the oxygen, the organisms can be classified into 5 types. The first one is obligate anaerobes. So usually these organisms, especially these prokaryotes, they rely on energy generated from fermentation or anaerobic respiration. Actually, they don't need any oxygen for their metabolism, and if some oxygen is present in their environment, it will affect their growth. What is the reason? The main reason is they lack mechanisms that protect against what can we say, reactive oxygen species produced by oxygen. Why there is a need for protection from reactive oxygen species? Can anyone tell? Like, we are also living in an aerobic environment only. Do we have the mechanism to protect against oxygen? Protect ourselves against oxygen? Can anyone answer? Do we have any certain mechanisms to protect against oxygen? Yes, we have. So that's why we can survive in aerobic environment. Actually, oxygen is so reactive. It can react with n number of entities. So without any protective mechanism, we cannot utilize oxygen. The same applies with the microbes also. So if certain microbes lack the mechanism uh, for defending against the attack of reactive oxygen species, we can call those microorganisms as anaerobes. They can only survive in absence of oxygen. So we can call those microorganisms as obligate anaerobes. So they can survive only in the absence of oxygen. Aero tolerant, the second category of organisms are like aero tolerant anaerobes. These aero tolerant organisms usually survive in an anaerobic environment, but they can tolerate the oxygen. They have certain protective mechanisms to prevent the damage against oxygen radicals. So these organisms are termed as aero-tolerant anaerobes. These anaerobes rely on fermentation to gain energy, and they also have certain protective mechanisms so that they can survive in oxygen containing environment or aerobic environment. But basically they are anaerobes. So these organisms are termed as aero-tolerant anaerobes. Facultative anaerobes. Actually these anaerobes can generate energy through fermentation and also by aerobic respiration. So that these organisms are called as facultative anaerobes. They can survive in both the environment and the mode of nutrition also. They can adapt to the oxygen containing environment and also the anaerobic environment. So these organisms are termed as facultative anaerobes. Coming to the microaerophilic bacteria. So the for these bacteria to survive, the oxygen should be present, but in very less concentrations. So these bacteria are termed as microaerophilic bacteria. So for this bacteria to grow, oxygen should be present but at very low concentration, that too in the dissolved state. So these bacteria are termed as microaerophilic bacteria. Obligate aerobes need oxygen to survive. So they cannot survive without oxygen. So these microorganisms are termed as obligate aerobes. So coming to the aerobic respiration, if oxygen is not serving as an electron acceptor, then what are the other entities that facilitate the electron acceptance? Usually microbial metabolism relies on the electron transfer reactions. In the case of aerobes, oxygen plays the role of electron acceptor. Then in anaerobes, what are the entities playing the role of oxygen? That is electron accepting role. So it differs with respect to different organisms. In the case of denitrifying bacteria, nitrate and nitrate play the role of electron acceptors. In the case of sulfate reducing bacteria, sulfate ions plays the role of electron acceptor. In the case of sulfur reducing bacteria, elemental sulfur plays the role of electron acceptor, and ferric ions or the Fe3 plus ions are the electron acceptor in the case of ion reducing bacteria. So these inorganic substrates or inorganic electron acceptors will vary with respect to different organisms. And finally methanogens use carbon dioxide as electron acceptors. So basically in anaerobes, the electron accepting moieties will change, with respect to the kind of metabolism they are adapting. So the next class of organism will be covering is fungi. So the fungi has a cell size in the range of 5 to 20 micrometer. Basically, they are larger than other prokaryotes, which are nothing but bacteria and archaea. So these usually fungal cells are organized into filaments. These filaments are called as hyphae, and this hyphae collectively forms mycelium. So fungi are utilized in different applications. So these are the applications in which fungi are used. Basically fungi are used for bioremediation. So these fungi can consume xenobiotic compounds and detoxify the environmental matrices like soil, air and water. So basically fungi are used in bioremediation to detoxify the environmental matrices. Other than that, fungi are used in agriculture. So have you heard about mycorrhizal fungi? Hello. So this mycorrhizal fungi used to grow at the bottom or near the roots of plants. So it will facilitate the uptake of nutrients by converting the organic matter into utilizable form. And these fungi also plays a symbiotic relationship with different plants. So in this way fungi helps in the agriculture. In food industry, fungi are utilized for producing different fermented products like soy sauce and beer. Other than that, other milk based products are also being produced by fungi, for example cheese. For baking also, yeast are being used for leveraging, what can we say, for producing bread and other baked items. In medicine, for producing antibiotics, immunosuppressants and other pharmaceuticals, fungi are being used. Then fungi are also, the enzymes derived from fungi are also used in different industries like textile industries and paper industries. And fungi are also used as model organisms to study different since fungi are a eukaryotic organism, we can correlate our fungi with the human systems. So these fungi are used as model organisms specifically to study the genetics and cellular processes which are associated with the different diseases. So in this way fungi are being useful. So basically fungi are classified based on their life cycles. And the presence or absence of fruiting bodies and the arrangement of and type of spores they produce. So if a fungi has interconnected filaments and if they have interconnected filaments, they can be called as molds, like especially multicellular filamental molds. We can see these kind of fungi in rotten bread and other rotten food. So the filamentous fungi are termed as filamentous molds. Basically they are called as molds. Then macroscopic filamentous fungi, they can have large fruiting bodies. So mushrooms are the best example for macroscopic filamentous fungi. So the visible part of mushroom can be termed as fruiting body. So these mushrooms are also coming under the macroscopic filamentous fungi. So finally, single-celled microscopic yeast. So these yeast are used in different processes, as we know, for fermentation and for producing different beverages. These yeast are being used. So this is the classification of fungi. Basically, fungi are classified based on the life cycle and the kind of fruiting bodies they are having, and also the arrangement and type of spores they produce. So fungi can be classified as multicellular filamentous mode, macroscopic filamentous fungi, and single cell microscopic ones. So this classification is based on the like fruiting bodies and the structure of fungi. So based on the conditions, we can call or classified fungi as oomycetes. So if a certain fungi is present in water or moist environments, those fungi are called as water molds, and in scientific terms we can call those fungi as oomycetes and zygomycetes. Usually these zygomycetes or zygospores are produced during sexual reproduction. Ascomycetes refers to like sac-like structure of fungi and yeast. So these ascomycetes are capable of producing spores called conidia. And basidiomycetes, club fungi and mushrooms are brought under the class of basidiomycetes. So usually they have club-shaped structures. So that's why they are termed as basidiomycetes, or which means the fungi has a club-shaped structure, and deuteriomycetes. So these deuteriomycetes doesn't have any sexual reproductive stage, proper sexual reproductive stage, and these fungi are termed as deuteriomycetes. So molds, yeast, and mushrooms are different classes of and distinct types of fungi, and they vary with respect to the different characteristics. In the case of molds, these molds are like filamentous fungi. So they have mycelium and produce asexual spores called conidia for their reproduction. And these spores can also act as allergens and may create health problems to human beings. So basically, molds are filamentous fungi. And coming to the yeast, yeast comes under the class of ascomycetes. These are like single-celled fungi. Usually these yields reproduce through budding or binary fission. So in budding, the yeast will specific yeast will produce buds. These buds will separate from the yeast. So this mode of reproduction is called as budding reproduction. In the case of binary fission, one yeast cell will be separated into two daughter cells. So this kind of reproduction is termed as binary fission. In the case of a single yeast in the case of budding, a single yeast can produce multiple yeast through producing different multiple buds. But in the case of binary fission, only two yeast can be produced due to fission. Other than that, mushrooms are mushrooms are also filamental fungi, but they have large fruiting bodies. In that way, they differ from the rest of the two other classes, molds and leaves. So these mushrooms have hyphae, which are located below the ground. So the next class of microorganisms are algae. So usually these algae are floating eukaryotic microorganisms, and they are autotrophs, and they obtain their energy from light. And the size of algae also lie in the range of 10 to 20 micrometer, similar to fungi. So these algae, as we discussed, they carry out oxygenic photosynthesis, which means oxygen is involved in the photosynthesis, and also based on the pigmentation, the algae can be classified. Usually the algae has chlorophyll pigment in it, so mostly green colored algae are like predominant, but other colored algae are also there. In that, other different pigments will be present. For say brown, yellow and red algae are there. In that, chlorophyll also be present. Along with chlorophyll, some other pigments will also be present, which bring the different colors to the algae. For say brown, yellow and red. Mostly we can find algae in the aquatic environments and in the soil surface. Usually cyanobacteria will also have a blue-green appearance, so we should not confuse cyanobacteria with the algae. So there are like lot of differences between algae and cyanobacteria. So algae comes under the class of eukaryotes, but cyanobacteria comes under the class of prokaryotes. So in this way, algae and cyanobacteria are completely different, even though their appearances are somewhat same and confusing. These entities are completely different based on the karyotype. Yeah. So as we already discussed, the classification of algae is depending on the type of chlorophyll content and also the other pigments. So we can classify algae as chlorophyta, which is nothing but green algae. Then chrysophyta, which refers to golden brown algae. Euglenophyta. Why euglenophyta? Because they have no defined cell walls. So these fungi, not fungi, these algae are classified as euglenophyta. Then pyrophyta, dinoflagellates, which means diflagellates. These pyrophyta has dual flagellates, though that's why it is called as pyrophyta. Then rhodophyta. Rhodophyta means red algae. It refers to red algae. And phyophyta, which refers to brown algae. So based on the color, basically we are classifying algae based on the chlorophyll content and the pigment present in them. So based on that, we can classify algae into 6 classes: chlorophyta, cryophyta, euglenophyta, pyrophyta, rhodophyta and phyophyta. And the next class of organism is protozoa. So these protozoa are unicellular organisms. They can ingest and digest organic matter, and these protozoa lie in the size range of 10 to 50 micrometer. These protozoa are somewhat comparatively bigger than algae and fungi, and some of the protozoa are pathogenic. So before consuming any water, we should ensure that it is free of protozoa or any other microorganisms. So As we discussed earlier, these protozoa are unicellular organisms. They survive by ingesting other microbes or macromolecules. So that's why they are having a big cellular size, 10 to 50 micrometer, so that they can ingest other microbes and survive. So basically, protozoa has a bigger cellular size, which is for consuming the other microbes. So it gives the protozoa upper hand in terms of size, so that they can consume on other microorganisms and macromolecules. So usually these protozoa tend to form cysts at unfavorable conditions. So these cysts are resistant to desiccation, which is nothing but moisture less condition, starvation, nutrient less condition, and high temperatures and disinfection. So this cyst can survive a wide range of environmentally unfavorable conditions. So based on the mode of development and mode of movement, so not development, based on the mode of movement, protozoa can be classified into amoebas, flagellates, ciliates, and non-motile protozoa. Usually amoebas has no provision for their movement, and it also has pseudofeet or pseudopodia, and these protozoa are termed as amoebas. In case of flagellates, they have flagella. These protozoa has flagella for motion. So these protozoa are termed as flagellates. In the case of ciliates, cilia helps in the locomotion. There are also non-motile protozoa which has no provisions for locomotion. So if there is some changes in the protozoan community, which indicates there are some changes in the aerobic wastewater treatment. So we can use this protozoan community as an indicators for change in different aerobic wastewater treatment parameters. So if the population of amoebas are high in a particular water matrix, we can say that water matrix is enriched with organic matter, because these amoebas tend to grow in an environment where organic matter is present in more amount. So which can be correlated to the BOD, high biological oxygen demand. So if the amoebas are present in high amount, we can correlate that with the presence of high biological oxygen or biological or biochemical oxygen demand. So if flagellated protozoa or free swimming ciliates are present in a particular water matrix, and then we can correlate that with the presence of So if flagellated protozoa and free swimming ciliates are present in a particular water matrix, we can correlate that with the presence of high microbial, not microbial, specifically high bacterial communities. So basically we can correlate this with activated sludge and moderate BOD levels. The BOD levels will be moderate in the case of flagellated protozoa and free swimming ciliates. So protozoa also plays a major role in reducing the bacterial populations, specifically pathogens. These protozoa can consume the pathogens, and by consuming these pathogens, it will make the activated sludge process can less hazardous. So here protozoa plays a major role by consuming the pathogens. So other than that, stocked ciliates are also there. So if they are mostly they can be found in water matrix, which are low in bacterial concentration and biological oxygen demand levels. So how protozoa are indicating different parameters. So amoebas, if amoebas are present in particular water matrix, we can say that that water matrix is rich in organic matter, and also that water matrix has high biological oxygen demand. Flagellated protozoa and free swimming ciliates are present, then we can say that the BOD levels will be moderate in that particular water matrix, and the bacterial concentrations will be very high. In the case of stocked ciliates, these stocked ciliates will be mostly found in low bacterial concentration and water matrix with lower biological oxygen demand. Finally, helminths. These helminths are nothing but parasitic worms, mostly commonly found in humans and animals. So these parasitic worms like survive by consuming the nutrients from humans and animals. Usually these helminths tend to form microscopic cysts, which are referred to as seeds. So usually we should develop a environmental engineering system that should inactivate the cyst in water, wastewater or solid waste, so that these helminths will not be transferred to human and animal systems. So we should develop an environmental engineering system which ensures that these cysts are inactivated. So these helminths are also the cysts created by these helminths are also hydrophobic in nature. The surface of cyst will be hydrophobic in nature, so they can accumulate in wide range of matrices like landfill leachate and foam generated from aeration tanks or floats of sewage and other primary treatment. They can find in those hydrophobic surfaces due to the hydrophobic nature of the cyst. So finally, coming to the modeling. Before modeling, we should know modeling of microbial system. We should know the composition of a biomass. Then only we can correlate the. Hello.
Sir,
Yes.
I have a question.
Yes.
Sir, we saw that helminths when engineered they are used for restorative treatment. So
No, not
in
helminths
natural-
helminths are not engineered here. We should develop a environmental engineering system so that this inactive cyst, right? So this inactive cyst can cause diseases in humans and animals, parasitic diseases. So in order to avoid this, we have to ensure that this environmental engineering system should inactivate these cysts. That's what we were discussing.
So yes, sir. So for restorative treatment, we should use helminths, or they are caused by helminths.
Actually, in wastewater treatment, we should ensure that these helminths are removed. That is the point here.
Okay, sir.
Yeah. Actually, this helminths has no role in water treatment. But
So, sir,
these helminths are
which-
harmful to humans and animals. So we should ensure that these helminths are removed from the water.
Okay, sir. So which organisms should be used for wastewater treatment?
For wastewater treatment, we can use fungi, algae, bacteria. Yeah.
Okay, sir. Okay.
Yeah.
Okay. Thank you.
Helminths cannot be used for wastewater treatment. Yeah, thank you.
Okay, sir. Thank you.
So actually, we should develop a wastewater treatment system that inactivates cyst formed by helminths, so that they won't be transmitted to humans and animals. That is the point here. So yes, in order to model a particular microbial system, we should know the composition of the biomass. Why is it necessary to know the biomass composition? Can anyone tell that? Why is it necessary to know the biomass composition?
Might be to study the metabolism of any microorganism.
Yeah, metabolism we need to study. So for studying the metabolism, what are the factors we should be knowing? Basically, mass balance. We should know the mass balance. What is going inside and what is coming outside, isn't it? Hello.
Yes, sir. Yes, sir.
Yeah.
So the nutrients they are uptaking.
Yes. So in that case, we should know the composition of biomass. So then only we can do the mass balance. How much amount of nutrient is converted into biomass and how much amount of energy nutrition is converted into energy. So to have an idea about this, we should know the composition of biomass. So basically, biomass composition is represented as CH 1.8, O 0.5 and N 0.2. So this is the general representation of biomass composition. So basically, microbial biomass contains about 50% of carbon. So how we can empirically determine the composition of biomass? So there is an instrument called COHN analyzer or CHNS O analyzer. So with the help of that instrument, we can tell the elemental composition of biomass. So we can also use that instrument to tell the empirical composition of biomass. So here, if we take the growth of aerobic growth of a biomass, which utilizes carbohydrate as substrate. So this carbohydrate can be converted into, can be utilized to for different applications. This carbohydrate can be utilized for different applications. The first one can be utilized for growth. A certain part of like what can we say, oxidized carbohydrates can be utilized for growth, and a certain amount of carbohydrate can be utilized for the reducing equivalents for the synthesis of biomass, and certain amount of carbon can be utilized for assimilation of the elements into biomass. So this is how a carbohydrate is utilized. Usually the carbohydrate can be converted into energy, directly converted into energy, or it can be converted into reducing equivalents, or it can be converted into biomass components. Basically, a carbohydrate can be converted into three ways. It can be utilized for producing energy, and which will be utilized for the maintenance of cell growth. Then it can be used for generating reducing equivalents, which are nothing but H plus ions, or it can be utilized for synthesizing biomass components. So in this way, carbohydrates are utilized. Usually microbial biomass has 70% of water, and 30% of cellular materials. This is a rough calculation. So usually there will be slight variations will be there, but this will be the majority of the case. So in the case of dry cell weight biomass, it is constituted of different macromolecules, RNA and DNA. So the dry cell biomass is constituted of macromolecules, RNA, DNA, and this and other cellular components. So this dry cell biomass will vary with respect to different organisms. How it will vary? In the case of algae and fungi, the cell growth will be very low than comparing to the prokaryotes. So these cells, especially algae and fungi, will have cell wall materials and other kind of inorganic materials as a dry cell weight. The majority of components of these cells will be cellulose, silica and calcium carbonate. Since they have low growth rate, the RNA and DNA content will be very less in these organisms. But in the case of prokaryotes, the RNA and DNA content will be more comparing to the algae and fungi. But in the case of algae and fungi, due to their slower growth rate, other components will be prevalent like cellulose, silica and calcium carbonate. So basically the components of a microbial biomass can be determined by this equation. Here small ca refers to the overall content of the microbial biomass. So here capital Ca refers to the average quantity of component I in the cell. This component can be any component like cellulose, as we discussed in the previous case, silica and calcium carbonate. So Ca is the average quantity of component I in the cell, in a cell, whereas C1 refers to the average quantity of components, which depends on the growth rate and medium conditions, specifically DNA and cell wall material. So these components are weakly depend on growth rate and medium conditions. So and C2 refers to the average quantity of components which are more depend on specific growth rate. Here RNA and protein comes under the C2 components. So finally, this C2 components will vary with respect to the growth rate, and C1 it doesn't depend on the growth rate and medium conditions. So here we are not multiplying with any factor, but in the case of C2 it depends on growth rate. We are multiplying it with the growth rate, and C3 which is nothing but the storage components, which depends on the medium conditions. So we are multiplying with the factor n. This factor can be derived from the medium conditions. So the component of any cell can be derived from this equation. Basically we'll be dividing the average quantity of component I in the cell by the average of components of cell, which depends on growth rate, and the average components of cell which doesn't depend on growth rate, and also the average quantity of storage components in a particular cell. So if we divide all these components with the overall content, will get the content of any component of a cell, of a microbial cell. So for growth of a microbial system, um, growth medium plays a major role. So the growth medium can be defined medium or complex medium. So in the case of defined medium, the chemical cell components are known. We'll be externally adding the components, and we'll be knowing the composition of the components we are adding. So in that case, we'll be calling the medium as defined medium. So this defined medium is basically used to culture photolithotrophic autotrophs such as cyanobacteria and photosynthetic protists. Here photolithotrophic autotrophs refers to organisms which rely on light for synthesizing their own food, and lithotrophs refers to inorganic substrates which they consume. So basically photolithotropes can be cultured using defined medium. So basically defined medium is used in research. So where the experimental organism, microorganism is well understood and the metabolism is well understood. So in that case, we'll be using defined medium. In the case of complex medium, the chemical composition of the complex medium will be unknown. So actually there are lot of complex media are there. I heard about yeast extract, peptone extract. So those are comes under the class of complex medium. We don't know the exact chemical composition of those entities, and so these we call those medium as media as complex media. Basically these complex media are rich in nutrients and can be used for a variety of for growing a variety of microorganisms. So basically why we need a complex medium? So if we don't know how a particular microorganism is metabolizing or how a microorganism is growing, in that case we can use a complex media. So since this complex media has a number of nutrients, they can facilitate the growth of unknown microorganism also. So that is the purpose of complex medium. In the case of defined medium, we'll be adding the chemicals and we'll be knowing the composition of each and every chemicals. So mostly only if we know about a particular microorganism and we know how a particular microorganism is metabolizing, in that case we'll be using defined medium. In the case of complex medium, if we are unsure about a particular organism, then we'll be using complex medium. And we also don't know the exact composition of complex medium, and the complex medium is also rich in nutritional requirements. Yes. So cell growth can be in order to model a cell growth, we should study how cell growth cycle is happening. So growth of individual cells involve growth, proliferation and differentiation. Growth refers to increase in size of the cell, proliferation refers to increase in number, and differentiation refers to differentiation of a particular cell into individual cells. So basically growth of individual cells involves growth, proliferation and differentiation. Yes. So ultimately this cell differentiation facilitates the growth of microbial cell into specialized cell. So for example, endospores. We'll be discussing in the upcoming slides about endospores. So these are some classes of what can we differentiated microbial cells. So endospore is one of the classes. So it is nothing but endospores are less in water content, and it also surrounded by a protective coat around them. Basically, this endospore forms a mother cell, which what can we say, which can survive under harsh conditions like starvation, dry environments, and high temperatures. This mother cell will protect the endospore from the harsh conditions. Basically, during unfavorable conditions, this endospore is formed. And another class is exospore. So this exospore is similar to endospore, and the only one difference is the exospore is formed outside the mother cell. In the case of endospore, it is formed inside the mother cell, whereas this exospore is formed outside the mother cell, and the physiology will be similar to endospore. The physiology of exospore will be similar to endospore. And another differentiated cell is anabiotic cyst, which is nothing but enlarged dormant cell, which is designed for surviving under, this is also designed for surviving under unfavorable conditions. And another class includes nitrogen fixing cysts and bacterioids. So these are nothing but enlarged cells. So they are also capable of converting atmospheric nitrogen into organic nitrogen compounds. So these are some of the classes of differentiated microbial cells. So phases of cell cycle in the case of microbial system, the cell cycle phases can be classified into G1 phase, S phase, G2 phase, and M phase. So G1 phase is nothing but the period between cell division and initiation of DNA replication. Actually, cell division is the last phase of cell growth, and initiation is the first phase of initiation of DNA replication is the first phase of cell growth. So between these two stages, G1 phase occurs. So have you heard about mitotic cycle and meiotic cycle?
Yes, sir.
So mitotic cycle, what does mitotic cycle refers to? Non-sexual cell division, mitotic cell division, isn't it? Mitotic cycle refers to non-sexual somatic cell division. Simply to mention somatic cell division. So the duration of this mitotic cell division is usually proportional to the duration of G1 phase. Then the differentiation of cells from G1 phase starts here. So after that there occurs the S phase. During S phase, chromosomal DNA replication happens. So during the G1 phase, the initiation of DNA replication starts, and in the case of S phase, the chromosomal DNA replication usually happens. And in the G2 phase, which is nothing but the intermediate period between the termination of DNA replication and mitosis, the G2 phase occurs. After that, M phase occurs, which is nothing but mitosis, and during this phase, splitting of nucleus happens. So these are the 4 different phases of cell cycle. Usually the G1 phase occurs between cell division and initiation of the DNA replication, and the S phase occurs during the chromosomal DNA replication, and G2 phase occurs between the termination of DNA replication and the initiation of mitosis. After that, M phase occurs. So this is how cell cycle phases progress. Usually there will be coordination could be this cell division cycle and also the DNA replication cycle in a prokaryotic cell. So for this coordination, there are so many biochemical and physiological factors are involved in it. So usually there will be coordination between cell division cycle and the DNA replication cycle. So for this coordination to happen, there are multiple biochemical and physiological cell activities are involved. So which includes synthesis of RNAs and enzymes and their degradation, and regulation of enzyme activity by different metabolites and cofactors, and regulation of catabolism and energy storage, then finally regulation of whole cell activity by different cell regulators. So all these biochemical and physiological cell activities contribute to the coordination of cell division cycle and DNA replication cycle in a prokaryotic cell. So during cell cycle, 2 different nutrition types are observed: exotrophic nutrition and heterotrophic nutrition. So in the case of exotrophic nutrition, usually cells convert external sources of carbon into storage forms like glycogen, starch and lipids. So usually this occurs in the G1 and G2 phases of cell cycle. So what happens in the exotrophy? The external carbon will be stored in the form of glycogen, starch and lipids in the cells. In the case of endotrophy, the accumulated reserves of carbon reserves will be utilized for DNA replication and mitosis, and this endotrophy happens in S and M phases. So exotrophy happens in G1 and G2 phases, where usually cells convert external carbon into internal stored carbon in the form of glycogen, starch and lipids. And this exotrophy occurs in G1 and G2 phase. Usually G1 and G2 phases are called the cell cycle rest phases, and during these phases this happens. In the case of endotrophy, the stored form of carbon in the cells will be utilized for DNA replication and mitosis, and this endotrophy takes place in the S and M phases of cell cycles. So there are no regulations in the alteration of exotrophic nutrition and endotrophic nutrition in the uh, prokaryotes, but in eukaryotes there are some regulatory measures to alter between this exotrophic and endotrophic mutations. So basically those are determined by these cell signaling molecules like CAMP, which is nothing but cyclic adenosine monophosphate. So this signaling molecules enables the transition between exotrophic and endotrophic nutrition. So how we can mathematically relate the specific growth rate with the duration of G1 phase cell cycle and exotrophy. So actually these entities are related to each other and they affect the specific growth rate. So in this slide we'll be seeing how these entities are mathematically mathematically correlated. So usually the cell size, DNA content and percentage of exotrophic and endotrophic cells can be used to monitor cell population by flow cytometry. Usually if we know the cell size, DNA content and the type of nutrition a cell is undergoing, we can categorize the cells using a flow cytometer. Flow cytometer is nothing but an instrument which what can we say, which classifies or which counts the type of cells present in a particular population. So based on that we'll come to know duration of exotrophy. So how much time the cells are residing in exotrophy, and duration of G1 phase, and which are related to the duration of cell cycle, isn't it? So if we know the duration of exotrophy, usually when exotrophy happens, G1 and G2 phases of cells. So if you know the duration of exotrophy, you can calculate the duration of G1 phase, and which is linearly related to the duration of cell cycle T. So here TEX refers to the duration of exotrophy, and TG1 refers to the duration of G1 phase, and T refers to the duration of the entire cell cycle, and mu refers to the specific growth rate, and T refers to again duration of cell cycle. So and this duration of cell cycle is related to ln 2 divided by specific growth rate. So how this mathematical relation came? How this ln 2 by mu is related to duration of cell cycle? Basically growth rate is equal to ln 2 by time. So if you want the time of duration time, if you want the time of or duration of cell cycle, we can divide we can interchange these two entities. Actually growth rate is equal to ln 2 by t. So if you want the time or duration of cell cycle, you can bring this t here and bring down this specific growth rate here. So which translates or which can be mentioned as ln 2 by mu. So here if we see Tx, which is nothing but duration of exotrophy, can be related with this mathematical relation. Tx, which is nothing but duration of exotrophy, can be related to 0.5 factor of t, duration of cell cycle. minus 1. Here 1 can be considered as the entire cell duration, and if we subtract the factor 0.5 t, we will get the duration of exotrophy. And how this Tg1, which is nothing but duration of G1 phase, can be calculated: 0.7 of T minus 0.9. So if we bring this T to this to the left hand side, so, and if we bring the other factors at the right hand side, we'll get a correlation for T, isn't it? So if we equate this this expression with the already mentioned expression here, we'll be getting growth rate as is equal to ln 2 into 0.7 divided by Tg1, which is nothing but duration of G1 phase plus 0.9. So this is the ultimate equation or mathematical correlation we can use for the determination of specific growth rate. So if we know the duration of exotrophy and duration of G1 phase, so how we will come to know these timings? By flow cytometry. By flow cytometry we will calculate these timings. So with the help of these timings, we can calculate the specific growth rate with this mathematical expression. So initially we have discussed about the calculation of specific growth rate. Then how we can calculate the growth in a batch or continuous system? So how this growth rate will vary in the batch or continuous system? Basically, growth or increase in biomass concentration can be referred as X here, and proliferation or increase in number of cells can be referred to as N. So for a balanced microbial growth, there should be proportionate increase in the biomass and also the number of cells. So the number of cells is basically equal to initial cell concentration and number of generations. What is meant by generation here? What is meant by generation here? So generations are nothing but the same population of microbes present at a particular time, which reproduce to bring down the next generation. And N, which is nothing but the increase in cell concentration or increase in cell number, can be correlated with the initial cell concentration and the N, which is nothing but the number of generations. And the same applies to the growth of biomass. X can be correlated with the initial biomass concentration and the number of generations. And microbial growth has different phases. Have you heard about it? Lag phase, log phase, exponential phase. And at each and every phases, the microbial growth rate will vary. The specific growth rate will vary. So how it varies? It varies based on the variation of biomass with respect to time. Yeah. So we can write that expression as dx by dt is equal to mu x. Here dx by dt refers to the variation of biomass with respect to time, and mu refers to growth rate, and x refers to the biomass. So if we integrate these terms, we'll finally getting an equation which is nothing but mu is equal to ln x minus ln x naught by t. Here x and x naught refers to initial biomass concentration, and x refers to biomass concentration at a particular time, and by t refers to duration of the growth, exponential growth. So this is how we can calculate the microbial growth in an exponential phase. So how we can calculate the growth yield and growth efficiency in batch and continuous systems. So for a batch system, the yield coefficient or yield coefficient of microbial biomass can be given by y x by s, which is nothing but yield coefficient with respect to consumption of substrate. Yield coefficient of biomass with respect to consumption of substrate is referred to as yx by x. Here yx by s can be derived from the ratio of difference in biomass concentration to the substrate concentration, isn't it? Xt minus x naught will give the amount of biomass generated at a particular time, whereas S naught minus St will give the amount of substrate consumed. So this will give us the yield, how much amount of substrate is consumed and how much amount of microbial or biomass is biomass is produced. We can say how much yield is obtained, isn't it? This is for batch system. In batch system there won't be any interferences, so we can directly calculate the yield of biomass with respect to substrate with this expression. So in the case of continuous system, and considering there is no recycling of biomass, the yield coefficient can be represented as x by Si minus Ac. Here Si and Se are nothing but substrate concentration present in the influent and the effluent. In the continuous system, we will be continuously providing the nutrients to the microbes, so that the substrate concentration will be varying. So we have to consider the concentration of substrate in the influent and the concentration of substrate in the effluent. With that, we will be getting the amount of substrate consumed by the microorganism. And here x will be constant, y constant. In the case of turbidostat, the biomass will be constant, the concentration of biomass will be constant, but the concentration of substrate will be varying. So there will be only one entity, which is nothing but x. So we have to divide x by the amount of substrate consumed, which is nothing but Si minus Ac, to obtain the yield in the continuous system. In the case of batch system, we have to divide the substrate consumed with the biomass produced. But in the case of continuous system, here also the formula is same, but the factors will vary. Here Si refers to the substrate influent concentration and Ac refers to the effluent concentration of substrate. So this is how the calculation of growth yield and growth yield varies with respect to batch system and the continuous system. So batch culture is a semi-closed systems. Here we will be supplying the nutrients only once, and we won't be renewing the nutrient, and we also we won't remove the waste product generated also, but we'll be supplying air and gaseous products will be released out of the system. So that is why we can call this batch system as a semi-closed system, because nutrients and waste products are not removed from the cultivation system, but the air will be going inside, and the gaseous products will be coming outside of the batch culture system. So we can call this batch culture system as a semi-closed system. So how this semi-closed system or batch system is having an effect on growth phase? So due to after particular amount of time, the nutrients and nutrients will be completely utilized, isn't it? So it leads to accumulation of metabolites and biomass. So and this growth will not be facilitated further due to extraction of nutrients. So this is how batch cultivation is different from continuous cultivation. So let's see different phases of microbial growth. So then we can better understand the difference between batch and continuous mode of operation. So in the case of lag phase, usually the growth will be very low, or there won't be any growth in the lag phase. Usually what happens in this lag phase, the cells start to adapt themselves to the environment. So during this adaptation period, there won't be any growth. And this duration of lag phase depends on how much amount of biomass is inoculated, and the difference between previous and current conditions of cultivation. If the cultivation conditions are same, for say if a microorganism is residing in a particular environment, and we are providing the same environment, and this lag phase will be the time of lag phase will be very less. But if you are providing a completely different micro environment for an organism, then this lag phase will be extending, because this microorganism has to adapt to this new environment. So this will create an extension of lag phase. And the log phase will be short period. During this log phase, the growth of microorganism will be at the exponential rate. That's why this phase is called as exponential growth phase. And the transition period between log phase and stationary phase will be what can we say? The transition period, there will be certain transition period between log phase and stationary phase. After that, stationary phase occurs. And the stationary phase is characterized by very slow growth rate. This is due to the consumption of all the nutrients by the microorganisms. So if there is no nutrients, the growth obviously will slow down, and there will also be accumulation of metabolites. So finally, during this growth phase, the growth will be very slow, or it will be absent, and the growth will be maintained. That's why it is called a stationary phase. So due to the accumulation of metabolites at the stationary phase, there will be transition of growth phase from stationary phase to death phase. So this leads to increase in the number of dead cells, and also this is the space where the cell lysis happens. So what is the advantage with continuous culture? So continuous culturing is an open system, contrary to the closed system or the batch system we discussed previously. So how continuous culture comes under open system? Because the environmental conditions are maintained with the continuous provision of nutrients and waste removal. So we'll be continuously giving the nutrients, and waste will be removed continuously in a continuous system. So this makes the continuous culturing an open system. So what is the effect of continuous culture in growth phase? So the microbial population can be maintained in the exponential phase. So since we are supplying the nutrients continuously, the microbial population can be maintained in the exponential phase for a longer period of time. So only if the nutrients are exhausted, the microbes can enter the stationary phase. Or if the nutrients are continuously supplied, they will be maintained in the exponential phase. Yes. So by applying continuous culturing method, the exponential growth phase of microorganisms can be extended. That is the advantage of microbial continuous culture. Yeah, thank you. So during this lecture, we have discussed. Let us have a brief recap of the session. So we have studied how different omic-based approaches and how different disciplines can be integrated to study the process happening with the microbial communities, and what approach we should be relying on. We have learned about these things, and we also learned about how integrating different disciplines together helps in the development of NPTEL biotechnology. And we studied about the necessity for modeling and simulations. And so we studied about the necessity for modeling and simulation, and then we discussed about the challenges happening with the studying of microorganism. Then we discussed about the necessity for grouping the microorganism and quantifying them. And we discussed about the different classes of microorganism. Then we studied about how we can correlate biomass composition with the mathematical modeling, and why is it necessary to study the biomass composition. And how to calculate the components of microbial biomass. And we learned about the concepts of defined medium and complex medium and what are their uses. Then we went into cell growth cycle, and we learned about the phases of cell cycle and mode of nutrition happening during cell cycle, that is exotrophic nutrition and endotrophic nutrition, and how this exotrophic and endotrophic nutrition, the transition between these nutrition types are happening. Then we went to the mathematical modeling of specific growth rate, and we studied how this yield coefficient calculations varies with respect to batch and continuous system. And we discussed the difference between the batch and continuous mode of cultivation, and we also discussed the different phases of cell cycle. Then we discussed the advantage of continuous cell culture. Yeah, thank you. Any doubt in this session? Hello? we'll be wrapping up the session right now. If you have any doubts, you can.
Both nitrogen cysts and those heterocysts are all microbial differentiation, right?
Which one?
So those nitrogenous cysts and heterocysts when formed like to what to say for nitrogen as a
Cyst.
from the
You were talking
environment.
about cyst, right? Nitrogen, yeah.
Yes, yes.
So what is
they
your
all
doubt?
are micro.
Uh-huh.
So they all are all comes under microbial differentiation.
Yes.
Cell differentiation.
Yes. Cell differentiation. Yes. So
And endospore and exospores, they are.
those are also differentiations only. So in the case of endospores, these endospores will be formed within the mother cell, and exospores will be formed outside the mother cell. Are getting when the conditions are favorable, this endospores will come out and burst and will produce the progenies. And in the case of
Yes.
exospores, the spores will be produced outside the mother cells, and the conditions are favorable, the progenies will be released from the exospores. Only the difference is where the spores are present. If it is present within the mother cell, then we can call that as endospore, and if it is present outside, then we can call that as exospore. Structurally, this endospore and exospore spore are similar. Is that clear?
Yes, sir, clear. So cysts are microbial differentiation.
Yeah.
And endospores, they are also cell differentiation.
Yeah, differentiation.
Also, so what is the difference between cell proliferation and cell differentiation?
Yes. In proliferation, the number of cells will increase, isn't it? That is what the term proliferation refers to. In differentiation, what can we say that the cells will be differentiated into particular. It will attain some certain characteristics by differentiation. In the case of proliferation, only the cell number will increase, isn't it? But the cell characteristics won't be affected by the proliferation, but it will be affected in the case of cell differentiation. Are getting the point? The proliferation.
So cells get differentiated.
No. During the in differentiated, yes. In differentiation, the cells will attain certain characteristics. In the case of
Yes,
proliferation, only the number of cells will increase. The cells will multiply, but after multiplication, it should get some certain characteristics, right? That is facilitated by differentiation. So the proliferated cell can be differentiated into endospore or exospore or an anabiotic cyst or nitrogen fixing cyst and bacterioids. Are getting the point right now?
Repeat, sir, please.
Usually the proliferated cells will be for say what can we say, so those are microbes. Let's microbes are bacteria, so they will be in that form only. So after differentiation, they will be converted into endospore or exospore or anabiotic cyst or nitrogen fixing cyst. Now are you able to getting the point?
Yes, okay. So what comes under proliferation?
In proliferation, bacteria will be multiplied. Bacterial cells will be multiplied. So after multiplication, this bacterial cells, due to unfavorable condition, can be differentiated into endospore, exospore, or an anabiotic cyst or those kind of cyst and bacterioids. Now are you getting the point? During
Yes,
proliferation, only the number will increase. The bacterial cells will be the same. So based on the condition, those bacterial cells will be differentiated into endospore or exospore. If the condition is unfavorable, that bacterial cell has to be differentiated into endospore or exospore or any other form of different. It can be either cyst or bacterioids. So now are you able to getting the point?
Yes, sir.
Yeah.
Yes, sir. So in fungi mycelium, when conidia is transformed into conidial spores and So this is proliferation.
Yes, yes. No, no. Actually, that is differentiation. Proliferation is increase in the number of fungi.
Oh, the whole organism will
Ah,
multiply, right?
yes, yes, yes.
Okay. And the
If it
body
is
part when
if it is
you have
attaining
to.
certain characteristics, that comes under the differentiation.
Okay, okay. Thank you so much.
Yeah. So, any other doubt? So, if there are no doubts, we can close the session right now. Yeah, thank you all for staying during the session and clearing your queries. Now we can wrap up the session. Yeah. Thank you.
Thank you, sir.
Yeah.